Oh, yeah, I know.
Oops. I don't think I can fix that. That's not.
Yeah.
Oh, sorry, I didn't see that.
No problem.
Thanks.
Okay, so
What?
are we good? Are we ready to roll? Can people online hear?
Sound check.
You can hear, okay. Do you want to move this bar?
Yeah. Oh, yeah, I will need to move that bar.
You need to drag it.
Where do I get the mouse on here? Just are you
Also
see
turn
it?
the light off right underneath the board. Underneath this. No, talk. Oh, that way. There.
Oh, yeah. Let's get rid of that.
Okay,
Perfect.
that right there.
Great.
Then you'll never find it again.
I know. Exactly. Yeah, do it. Okay, well, welcome to our second IB Enlightening series. So today we're going to start with Chris Marshall, who's going to talk to us about the role of natural history collections and curation in 21st century science. And I'll also just mention that again, if anybody's interested in beers afterward at Downward Dog, we'll be headed there, hopefully with the speakers. And yeah, I'll introduce our next speaker after this.
Great. All right. Well, I'm going to get started right away because I have a few too many slides and I knew this, but I'm going to just try and speak pretty quickly. Thank you for the opportunity to speak today on a topic that I find really valuable and useful as my role as the curator for the Oregon State Arthropod collection, the insect collection here at the university that's managed by the Department of Integrated Biology. The reason I chose this topic is because I think all too often today, now my slide's not advancing here.
Click on the slide.
Go. All too often today, there's a common perception that natural history museums and the type of activities that take place in them are really kind of antiquated vestiges of 19th century biological sciences. And I can understand why that is. I kind of get it, is that in fact, the collections here on campus that I curate and other curators on campus curate have their origins back in the 1870s or 1890s. And in fact, these are pictures from the OSU archives showing the then zoological and entomological collections, which were combined into a single museum in Benton Hall with students, Victorian age students, frantically probably studying for their midterm. That makes sense too. The field of zoology and entomology at the time, museums and natural history played a center, kind of center stage within what it was, what we call learning outcomes today for our majors. But I'm hoping that over the course of this short talk today, I'll convince you that these collections on campus are not only maintained their relevance to biological science and inquiry, but they are incredibly important collections, both here on campus and elsewhere around the world. When I arrived here in what would be the earlier part of the 21st century, the collection was kind of in a frozen state and looked like something you'd see in the 1950s. I don't think anybody who works in Quarterly Hall would be overly surprised at that. And in fact, one of my first roles was to get the collection up to at least the basic standards that we would have for a late 20th century, early 21st century collection. And this is only one step along a long route that I hope someone will be here in 100 years talking about the value of this collection in the 22nd century. But we have actually replaced almost all of the World War II era wooden cabinets that were housing the collection. But the housing itself is only a small piece of what we mean by modernization. And in fact, all of the policies, the approaches to how we work with the collection down to the specimen level, as seen here on the left, has been modernized to modern standards. And of course, with the new quarterly renovation, I hope that sometime in the next year or two, when we finally get a chance to move back, I'll be having you guys there for an open house, and you can see our truly modern 21st century approach to an academic museum. We are not alone in our efforts to maintain and serve as a repository for arthropods and insects around the world. This is just a map showing a distribution, a kind of a draft distribution of all insect and arthropod collections on the planet. You can see that they're not evenly distributed, heavily biased towards Europe and Eastern North America. And in fact, if you zoom down into the Pacific Northwest, you'd notice that there aren't very many collections at all. In fact, here you can see there's us here on this little dot. You can see some other red dots there. The only other collections, if you took all the other collections in the state and put them together, we'd be an order of magnitude larger than them in terms of our capacity to store specimens. And in fact, we're at least twice the size of any other collection in the Pacific Northwest. So we do serve a really important role for providing scientists around the world with information about our local fauna. We have a number of strengths in the collection, mostly tied to the research agendas of various researchers here, but in general, we have good representation for insects and arthropods for most groups that you'd find here in the Northwest. There's a few exceptions. These are our strengths, however, which is the Coleoptera, the beetles, the Hymenoptera, the Hemiptera, the Lepidoptera, aquatic insects, and mites, known as the Acari. Those together make up about 75%, I would say, of our collection, but the rest, we have some great material in other areas as well. I'm not here to talk to you really about the strength of the collection in terms of what we have. I'm more here to talk to you about what we do. In other words, so you're 140-year-old and you have 3 million dead bugs, who cares? That's the question I want to address a little bit in my 15 minutes, or why should you care? As a quick little segue into this topic, I'm going to mention that it was interesting to me. I see collection work, whatever I look at. And 2 weeks ago during our lightning talks, 2 of the 3 outstanding presentations touched on museums and specifically touched on topics I'll be covering in later slides. First, David Madison gave a talk on his work to understand the radiation of bembiodines from a little island called St. Helena. And he was only able to accomplish that work because of the museums and museum specimens that were made available to him to other institutions. And secondly, Dee Denver gave an outstanding talk on Minikata Kumagusu, and this highlights another really important element and facet about natural history collections in general, is we are a hub for natural history researchers, scientists, community members, and a diverse assemblage of people to engage with each other about biodiversity and to contribute important observations and specimens to the endeavor. So I want to kind of abstract the idea of a collection for you a little bit in terms of what they are and compare it to 2 things that you're very used to seeing around laboratories today, the microscope and the telescope. Maybe not the telescope so much in the biological sciences, but the microscope and telescope have similar like old, centuries old origins in the field of science and their utility. But what I point them out as is because microscopes allow scientists to basically take complicated and difficult patterns and phenomena that take place at a scale that we simply cannot see and make sense of without the use of tools. Telescopes likewise allow scientists who study the cosmos to look out and examine phenomena that take place at scales much larger than we are. And I'd like you to consider the idea that natural history collections are in fact a type of research tool. So don't think of them as drawers of bugs, but think of them as an actual facility that allows scientists to examine biodiversity at scales that we simply cannot do for a single individual. Ecological studies can be done if you go out in the field, you do an experiment. Biodiversity studies in terms of species distributions over long-term time periods, historical time periods, simply cannot be done without access to a wide array of specimens. Taxonomists have always known this. In fact, taxonomists are the heart of many of the museums, and that's definitely the case here at Oregon State University in our collection. And we use these collections in order to gain enough information about species across their distribution and over time to basically describe the biodiversity around us. With regard to insects, this is incredibly important. This is just a graph from a work that looks at the relative proportion of described species on planet Earth based on what's known about them, as well as what we know in terms of any sort of sense of natural history or actual biology. And what you'll see here, or what I hope stands out, is how many undescribed insects are there. They're not the only poorly represented group on the planet. Certainly mites and nematodes and a host of other organisms are poorly represented and understood by scientists, but insects are a pretty large one, and they're also a pretty important one for terrestrial and freshwater ecosystems. So why is that important? Well, species are disappearing at an ever greater rate today. And so I kind of feel like maybe the insects is going to be the sequel to like, don't look up, it's don't look down. And we basically, people just don't understand what's disappearing. Many large-scale studies are looking at the insect apocalypse, and some are coming down one way or the other way, but it's pretty clear no matter how you slice it, in various systems, the diversity and the abundance of insects in a lot of these systems is simply declining. And that's a problem if we don't understand the historical records. And museums offer the opportunity to examine these distributions back in time. So that's not surprising that the role of the insect collection here at Oregon State University has played a greater role in working with various land managers and private and public institutions around the Pacific Northwest to help provide them with historical data for these sort of species of concern or that are threatened. We provide the vast majority of information, in fact, on some of these species locally, and we support the researchers who do the surveys associated with monitoring them every year. A very recent project I want to bring to your attention is a collaborative project that the Oregon State Arthropod Collections is involved with members of the Department of Horticulture and Oregon Department of Agriculture. It was discovered a few years ago, well, not discovered, but it was realized that the knowledge base for understanding the pollinator services provided around the Pacific Northwest was simply not knowable. We didn't have enough baseline information available to understand whether or not our native bees were in decline or not. With a successful grant, federal grant, we were able to basically begin the process of rectifying this problem. There were funds available to hire students to work in the collection to digitize large portions of the bee collection, and a current grant that's submitted right now would allow us to complete that. In addition to this, OSU Horticulture has been engaging and designing a gigantic, massive army of bee survey work around the state, and all that material is being combed through by taxonomists, and then selected material from that is being deposited in the Oregon State Arthropod Collection. We're also the institution that's taking all of the observational records and the specimen records and making them available online through portals like GBIF. This is just an example of the 2018 data set of bees around Oregon. But that's only one of a few of our massive collaborative digitizing efforts. We just finished a collaborative project with a number of other institutions around the country where we digitized Lepidoptera, our butterflies and moths. I already mentioned that was one of our strengths, and in fact, we provided data for a lot of species and taxa that were not available in any of the other institutions. We provided 97,000 data records. We still have another 40 to 50,000 records to add to that. And why is that important, and what does that allow scientists to do? Well, just this last year, other researchers mined that data off of GBIF from all of those collective institutions, and they were able to basically begin to answer questions about broad-scale landscape changes and diversification rates and associate those with the geospatial information available through those label data and historical. Really incredible stuff. But that just scratches the surface because, as most of you are aware of, some part of your laboratory probably touches on some form of a next-gen sequencing molecular techniques. These are just now finally reaching a stage where we can actually get material, DNA material, out of museum specimens, and that's something that was highlighted in David's talk. But this is being done at a much larger scale, and people are using entire museums to basically find out information about the flora and fauna within their communities. In addition to the target DNA of the organisms in those collections, with new eDNA metabarcoding techniques, we're also able to utilize those same approaches to understand more about the organisms that live in, with, and along with those creatures. In addition to using isotope analysis, we can use the specimens to understand things about the physical attributes, biogeochemistry of the environments they were inhabiting. What does this mean for researchers who study these things? It means they can now basically take their studies and look at them in a historical context, which really gives them some strong power. So, natural history collections are involved in all this research, but now this is where the comparison to microscope and a telescope falls apart. Natural history collections are not mass-produced objects that you can buy on a regular scientific catalog. They're basically gigantic, long-term collective endeavors built by individuals, by people. They're essentially more like libraries than they are like a microscope. So while you can have a collection in your lab and you might find a lot of use in it, the real value of a natural history collection is the fact that it's an aggregated resource created by the multiple collections of different researchers pooled together, and that provides a resolution and a capacity to examine questions beyond the scope of your individual lab. People are important in maintaining these collections because, again, unlike a microscope, if they're not utilized, not only do they become degraded and broken over time through damage, but they can also become damaged just by poor or inappropriate use. And they essentially become irrelevant. They cease to be able to address questions about contemporary biodiversity. In some regards, I was mentioning earlier that this talk could be something for the sciences of the 22nd century. In fact, we're thinking about that. How do we basically build the resource so that it's something useful for somebody addressing questions now? Curators are involved in building the collections and making sure the material that shows up is both accessible, has information that's useful for researchers, and is prepared in such a way that it can be saved for long-term purposes and easily found and the information extracted. We do that by encouraging gifts, donations, research deposits of vouchers, and we also do our own field work. This is why, in fact, you'd see the strength of the Oregon State Arthropod collection, and many of the collections on campus reflect the research interests of the curators who are involved in the management of the collection. These are just 3 examples of beetles that I work on. We have outstanding collections of them already, but I'm also adding more material to them as I go. David is doing the same thing with regard to his carabid interests. Lastly, I want to point out that these natural history collections are regional biodiversity hubs. It's really important. It can't be understated how much they serve as a physical location, a place where an oasis, so to speak, where researchers interested in natural history, taxonomy, systematics, and biodiversity in general can congregate. These collections serve as a place where our local biodiversity, our insects and arthropods, can be shared with researchers around the world, both by them visiting us and by processing of loans, which I do a fair bit of. We still do that in this day and age. And as well as a regional space for our local entomologists and local people who are interested in insect biodiversity can visit us, and they can see not only examples and a really good representation of our local fauna, but they get to see examples of those in relationship to other examples of insects from elsewhere around the world. Because we're an oasis and something that attracts researchers with a lot of taxonomic expertise, museums and collections are also associated with teaching, education, and outreach. So it's a natural sort of follow through of the endeavor. The expertise in the collection tends to be associated with taxonomy and biodiversity, of course, and that sort of information can be transferred to students, the public, and other individuals through a variety of mechanisms: courses, mentorship, workshops, outreach, and employment. This effort, this education, I found myself personally, I know it's true at the other collections on campus, really works well because it trains these individuals to be more effective volunteers and assistants in terms of curating the collection and basically improving the overall product. We still engage in outreach even though we're not on campus right now. So we do this in a number of different ways, but I did want to mention I'm putting a plug in for the Oceans 11 group and the clubs on campus, which are going to do a Dead Week tour. So normally I bring show and tell to show people actual insects from the collection, but I didn't this time. We're not going to have people in the collection, but we are going to have a Dead Week tour taking place in ALS on March 7th, 2022 from 6 to 10 p.m. This will be like the 4th or 5th Dead Week tour that's taking place on campus. So before I end, I want to quickly go back to this analogy so you can kind of think about it again before I give you, is that in fact, these natural history collections are incredibly unique and complex research tools that allow scientists to view and study large-scale geographic temporal patterns in biodiversity. That the curators and the people associated with those collections are critical, not just for the maintenance of those collections, but they improve and they connect those resources to the research communities, educational communities, and outreach opportunities that exist in the environment around it. OSEC in particular is a unique, irreplaceable, long-term collective endeavor that documents and makes available now and for the future scientists our regional and insect biodiversity. And I also want to point out, we talk so much about digitals, but collections stand in the face of that. We are fundamentally physical resources. And I think we offer a really great opportunity for people to engage in science in a hands-on, physical way. We still provide our stuff digitally. We're totally doing that, but we cannot be fully digitized. We are at our core physical objects in cabinets. And lastly, before I end, because the lightning talks were about introducing people, and I know a lot of people here don't know me personally, I thought I'd share just a few things about me. This is me and my husband and our new dog, Myra, truffle hunting. When I'm not in the museum, you might find me working on my marine aquarium or scuba diving someplace on vacation. I'm an amateur winemaker. I love gardening and orchids, knitting, and I'm a geeky Dungeons and Dragons player. So with all that said, I'm going to pass the baton to the next speaker because I know we're limited on time. Do we have time for questions?
Yeah, I think you do.
Great.
Let's do one question.
I know I talked fast, but I had too many slides. Yes.
Is the collection searchable online?
Yeah, that's a great point. So when I got here, we had no catalog, and we still do not have a searchable catalog. And that's a major initiative I want to undertake. In fact, we're starting that in our effort to move back onto campus. We're moving through, and I'm going to be hiring students to basically do a drawer by drawer inventory of the collection. So we'll be able to gross level be able to do that. Our other material is all done at the specimen level. So in order for us to participate in these large scale biodiversity projects, everything's done like specimen by specimen. But I started off when I first got here, I was including with that record what cabinet and drawer the specimen was located in, so you could go find it like a giant inventory, like a warehouse. But it became too impossible, and we kept shifting stuff, and we couldn't manage the actual data. So we gave that up in order to get more specimens available for biodiversity studies. But now we definitely want to have that. We want people to be able to search through the collection. But honestly, the way, if you want something in the collection, you need to rely on one of us to go get it for you, because there's no way to know if we have it really. Yeah, but good question.
Thanks, Chris.
Sure.
All right, the next speaker is Molly Bird. Take a minute to switch over the. Okay, so she's going to talk to us about evolution in a test tube. What can experiments with yeast teach us about the genetics of complex traits? And should we take that?
Oh yeah. Yeah.
All right. Well, thank you so much for being here today and for giving me an opportunity to talk about my lab's research. And it's so strange doing this without a mask on. So even though this is a short format talk, it's been a while since I've addressed our department. So I wanted to take this opportunity to give a bit of an overview of some of the projects going on in the lab, and also talk about our overall mission and why we do what we do. So a theme that connects all of the projects in the lab is really experimental evolution. And the organism that we work with most often is the yeast Saccharomyces cerevisiae. So what do I mean by experimental evolution? Essentially, what I'm talking about is research in which you bring a population of organisms, model organisms, into the lab. You subject them to some novel environment or only select individuals with desired traits, and then study what the evolutionary outcomes are at the level of the genotype and at the level of the phenotype. So these days, we generally sequence evolved populations and compare them to either control populations or ancestral populations to learn about what is the genetic basis of traits that have evolved over a period of time. And we generally don't just do this one time, we do this several times in parallel so that we can make statements that are generalizable about evolution as a fundamental process. And the ultimate questions that we're interested in addressing with this type of research that are enabled by this approach of experimental evolution are, number one, what regions of the genome can we associate with particular traits? So what are the genes or genomic features that underlie particular traits? And also, maybe more generally, what is the tempo and mode of adaptive evolution? In other words, from experiments like this, we can quantify evolutionary rates. We can learn about where adaptive genetic variants come from and what happens to them over an evolutionary time frame. So if you're familiar with this type of work at all, you've probably heard of the most famous long-term evolution experiment that there is, and that was started by Dr. Richard Lenski, who's a professor at Michigan State University, and he's sort of famous for, in 1989, coming up with this concept for an evolution experiment with E. coli. So with a single strain of E. coli, 12 of these flasks were inoculated, so 12 populations originating from one isogenic strain. And then they were just allowed to evolve since then. So, you know, Dr. Lenski's staff, students, postdocs, every day since 1989 have, you know, taken samples from these flasks and transferred them to new flasks continuously. And in E. coli time, that has amounted to over 75,000 generations at this point in time. And so for some context, we think that modern humans have probably been evolving for somewhere between 5,000 and 10,000 generations. So this is a really incredible resource in terms of learning about evolution. The trait, by the way, is a very simple one. It's just glucose limitation. So that's the stressful environment in this experiment. And I think that a couple of things stand out about this experiment that are really notable. Number one, it shows that evolution never really stops. So there's probably a Nature or a Cell paper that comes out almost every year from this group, despite all of these generations of evolution. So we continue to derive insight into how evolution works, despite this being such a long experiment. And the other thing that stands out is that they've created essentially a living fossil record with this experiment, because E. coli can be frozen indefinitely without dying. So this picture of Rich here is showing him with all of his freezer stocks. And these strains can be returned to. And so any intermediate time point can be revived from the freezer all the way back to the ancestor to restart an experiment or to, you know, ask a question about what happened over time. So, so you can see that E. coli is probably the best organism for this type of work. It divides in 20 minutes. You can get, you know, massive amounts of evolutionary time happening in a, you know, some PhD students' timeline. And also they're cheap and easy to maintain. You can freeze them, as I mentioned, but they have some serious limitations if we want to learn about evolution in a general way. So it turns out it's really important to study an organism that has sex if you're interested in these, addressing these questions about how evolution works, because asexually reproducing microbes evolve to novel environments fundamentally differently than sexually reproducing organisms like metazoans, like humans. So, the best model system that's a higher eukaryote that has sex is Drosophila melanogaster, the fruit fly. And there's lots of interesting work in the experimental evolution world that uses this organism, but it's not the best choice either because it has a 2-week generation time. So you're not going to be able to do those really long experiments with fruit flies the way that we can with microbes. And so that's why my lab has been taking this sort of hybrid approach where we have found a microbe that has sex. And so the yeast, Saccharomyces cerevisiae, it's the same, you know, the baker's yeast, the brewer's yeast, it's all the same stuff that we use in our food and drink. It's a fantastic model organism for genetics. And it has all these features that make it great for experimental evolution specifically. So it has these short generations, not as short as E. coli, but pretty short. Small genomes, you can freeze it, it doesn't die, but it's also a diploid eukaryote that can be made to, you know, under laboratory circumstances to shuffle its genome through sexual recombination. So basically all the experiments that go on in my lab make use of this organism in its sexually reproducing form. And I'm going to just show this and not really get into it, but I just want to point out that that's actually pretty hard to do. I mean, I'm making it sound like it's, you know, why wouldn't we do that? But it turns out it's tricky. And so most folks who do experimental evolution research with yeast don't bother with any of this because it takes some genetic engineering so that we have markers in the population that can be used to track the efficiency of sex and mating. And it takes a lot of different lab steps that impose selection pressures of their own to make sure that the yeast are continuing to reproduce sexually instead of mostly asexually, which is their default. So most of the sort of publications that my lab has put forward to date are actually about optimizing this process. So it is complex, but what I want to do next is kind of give a highlight of some of the, now that we have it working, give a highlight of some of the projects that have been able to make use of this system and what we have learned. So my first graduate student who got her master's a couple of years ago, Kislana Wing, was actually interested in studying the trait of being able to survive a freeze and a thaw cycle. So I said that yeast can be frozen and they don't die, but it turns out that's not completely true. And so Kislana had this idea that maybe if we start with a big variable population of yeast, not everyone, not all the cells will be equally able to survive a freeze and a thaw event. And so that's a complex trait that we can approach with our laboratory's methodology. And so sure enough, she found that only about 75% of the cells that we freeze survive the thawing process, but that this ability increases over time. So after 5 consecutive cycles of freezing and thawing, she was able to show that the viability following a freeze-thaw stress improves. And so what we could do is sequence the genomes of these freeze-thaw tolerant populations, and we can convert that into sort of a genome-wide map, where on the x-axis we have location along the 16 chromosomes of yeast. On the y-axis, we have a significance score or an association score. And we can see that there's really just one region of the genome that popped out as having significantly changed over time. And so all of the sort of genomic real estate on in this spot on chromosome 15, that's where we would look if we wanted to figure out, okay, what gene or genomic region might influence this trait of being able to survive freezing and thawing. And we can also zoom out on that region. So it looks really like a small region, but if you zoom out, it's not really that small. It's about 50 kb long, but we can ask, okay, well, of the variants that we find here, what do they do? Are they in genes? This region encompasses about 25 genes, so there's a lot going on, but a lot of them occur in one gene. It's called ALR1, or aluminum resistance, that has a known role regulating metal ion homeostasis across the cell membrane, but is not known to be important for freezing or thawing. Also, we can point to specific variants that might result in amino acid changes, so non-synonymous variants, such as this purple one, which occurs in the gene, which has this boring name, which we don't even know what it does. So in summary, what I'm saying that we can do with this type of experiment is we can uncover genes or genetic variants that were previously not associated with a particular trait that we can now associate with that trait. And furthermore, with respect to the mode and tempo of adaptive evolution, we can ask what exactly happened to these variants over time that were significant. So if we just look at the very top one, the one that changed the allele, or the variant that changed the most in frequency over time with this experiment, what happened was and there were multiple replicate populations in which we observed this, but the allele frequency went from about 30% to something closer to 60 or 70% over the course of the experiment. But importantly, it didn't go higher than that. So these alleles are not becoming fixed. And this figure might not be all that convincing of that point, but work done by others in the lab, particularly Mark Phillips, who's a former postdoc, but I don't have time to talk to you about all his cool work. This shows that this is really a common outcome that we see, that these adaptive alleles, they are maintained at intermediate frequencies despite really strong selection and long experiments, at least long in yeast time. So that's pointing to this fundamental property of evolution that's a little bit surprising. So adaptive alleles are not becoming fixed. Okay, so in my remaining time, I just want to, so that experiment is sort of, that's a full story, and I'm going to end this talk about more of a work in progress. So we've begun using our approach and lab methodology to tackle aging and longevity. And so you might be wondering, wait a minute, do yeast even age? That's, they're a single-celled organism. How do you quantify aging in an organism like that? And it turns out that yeast, when they're asexually dividing, they bud. And when they bud, the cytoplasm divides asymmetrically. So there's a mother cell. It's kind of hard to see here in this picture, but a mother cell will divide to form a daughter cell. And any sort of components of cellular damage remain with the mother cell. And then the daughter cell is fresh and new and doesn't have any of those hallmarks of aging. And another thing that's nice about yeast as a way to study this is they literally get marks of aging as they get older. So they get what are called bud scars that we can stain with fluorescent dyes because they're very chitin rich. So all of these little green donuts that you see are bud scars that we've been able to stain. And so every bud scar is the scar of a previous cell division. So this is a mother cell who's divided, 7 to 10 times. And over here, this daughter cell that's being born doesn't have any bud scars. So what we can do is we can use this bud scar staining technique as a way to select, in an experimental evolution context, select out the old cells and only let them reproduce. And so this is work that originated with my former postdoc, Ian Cooch, and now it's being, the one doing all the hard work is Katie McHugh, who's a current PhD student in the lab. And what we essentially do is we stain the cells, and Katie stains the cells, puts them in a cell sorting device, and we're able to pick out only the cells that fluoresce the most. Those are the ones that we think are the oldest cells, and discard all the rest. And there is this little blip at the very top. So this is a distribution of cells. The ones with the most fluorescence are usually dead because dead cells autofluoresce. So we don't include those, but all of the oldest cells get to contribute to the next generation. And our hypothesis is that over time, and this is based on work done in lots of other species and evolutionary theory, but our hypothesis is that over time we expect longevity to evolve as a consequence of this selection treatment. And we think that we have some initial evidence that suggests this is working. So Ian was able to do a pilot experiment with 3 replicates, showing that after about 6 weeks of this kind of a treatment, the amount of bud scars that we can measure in a given population is going up, at least in 2 out of the 3 replicates in this case. Whereas that pattern is not observed in control populations receiving identical treatment to the selected lines, except that we don't select for old cells. All the cells get to go through. So this is a pretty exciting result, but we need to, we're right in the middle of this research. So what we're doing next is we're repeating it because 3 replicates is not enough for this type of work. And also we need to do this for longer to see if those phenotypes shift more. We need to sequence the genomes of experimental and control populations to do that genotype to phenotype mapping that I mentioned. And also we've become much more interested in thinking about late life reproduction or reproduction late in life. In addition to just longevity. So the ability to reproduce when you're older is really what it takes to get through this experiment. You can't just be old, you have to be old and fertile. So what Katie has been doing has been working on using this selection experiment as a way to generate model, a laboratory model for late life reproduction. So I think that my time is up. So I will skip this last slide and go straight to my acknowledgements, and I'd be happy to take any questions if you have them. Okay.
So with the bud scars, that is effectively like the reproductive age, right? Does that correlate really well with like linear time, or is there a lot of variation how quickly they bud?
Yeah, that's a great question. So in yeast, you have to distinguish between replicative lifespan and chronological lifespan because replicative lifespan is what we're measuring. So the number of bud scars is how many times they can replicate, but that doesn't always, it correlates with sort of chronological time. But yeast can enter stages of sort of diapause or metabolic almost dormancy, where they can hang out for a long time without dividing, but we wouldn't consider that cell as aged as one that had many bud scars. So our trait of interest is replicative lifespan for that reason.
Yeah, that makes sense. That's the more important metric anyway. Are you doing like oxidative stress kind of stuff for aging or? Not
yet, no. I mean, this is still, we're just building the sort of
model.
Experimental apparatus right now. But yeah, the hope is that we generate this model with evolved postponed longevity and evolved postponed reproductive capacity so that we can test lots of questions about, why is it that fertility declines with age, right? In organisms such as humans. What are the hallmarks of aging in this model that are different from like the ancestral population mechanistically? So that's the goal, is to build the model.
This is just thinking about it as I see the problem. So this is mostly done in asexual differences, right? The aging work is mostly asexual, or
no, I mean
Okay.
they age when they're budding, but then in order to make it into the next generation, they have to undergo the sexual cycle.
okay, yeah. Because I was kind of curious if you were worried about mutation accumulation because of that kind of, but I guess the thought process.
Nope, I mean, it's the same sort of general machinery that I introduced, yeah. But yeah, there is a big bottleneck, so we need to be a little more concerned about things like that than we normally would. The sorting introduces a bottleneck that is a little bit different for us. Sorry, I think I saw Francis first and then Dave.
I just saw that Andrew, he's up to this database of like 7,000, evolution and natural system database. And I think it's just that the data is like change shift over time. But it doesn't, how far does it feel kind of find what you do to identify that fact that there's actually within the like, do you have those targets that you can then say, aha, let's look at what.
Sure, I see. Yeah, I mean, so any sort of genomic region, so yeast is a great model system because, the genes are homologous across the tree of life. So, right, so we could point to natural populations experiencing some kind of environmental shift and look for homologous genes or orthologous genes that might be relevant in that case. Does that answer your question?
Usually gets small sizes, age of reproduction decrease because of harvest pressure.
Yeah, well, I mean, you could imagine adding another dimension to this research where you control population size in a really specific way to test a hypothesis, which we've thought about doing. I think maybe one more question and then I'll.
Yeah, the first question kind of made me wonder, like, is there a way to definitively distinguish between reproductive age and like reproductively prolific yeast that just reproduce more frequently?
Yeah, right. So with any experiment like this, you're going to get like creative solutions that you didn't expect. So, right. So we've been worried about maybe the bud scars get denser and then the cells fluorescing more for that reason. Or if their growth rate accelerates, then you'll get more bud scars, but not necessarily a longer lived yeast. So those are all concerns for sure. But given that replicative lifespan is what matters to us, I think that outcome would be okay. So having a faster, growth rate, turnaround time, all that is still in line with our baseline expectations about aging and longevity and late life reproduction. But yeah, we're always looking out for ways that the cells are defying us. So thank you.
Thank you, Molly.
Fairly loud without this, but.
Okay, and our final speaker for the day is Anna Dulles, and she's going to talk to us about chasing the holy grail, resilience and disease ecology.
Right.
And just one last reminder that afterwards we will go to downward for the semester.
Sounds good. I can use my hands to point otherwise. No, it's fine. So I like to make interesting titles because they entertain my grad students and they get to make fun of me every time. So here we go, Chasing the Holy Grail, Consilience and Disease Ecology. So just as far as introductory material before this, there's two big projects going on in my lab. The most current one in terms of actual field work and sample processing and data acquisition is actually about desert bighorn sheep. And so this is a bit of a leap now. I'm going to be presenting some findings from our buffalo system which is beyond data collection, which is why it's ripe for presentation. But I just wanted to point out that those are both going on. Ooh, I'm gonna go with this one. So chasing the Holy Grail, well, there it is, the Holy Grail. And you know, there's very good reasons to chase the Holy Grail. It's very shiny and irresistible. And part of its irresistibility, though, is that you can never get to it. And I will be arguing here, along with E.O. Wilson, just in memoriam and paying my respects here, that the Holy Grail in most any discipline, and certainly in disease ecology, is a lot about connecting pieces that you weren't expecting to connect properly. It's about coming from different sides. And then the truly mind-boggling effect is when they come together where you couldn't really anticipate how that fit would work. And what's reassuring, though, and this is E.O. Wilson right here, is that if those committed to the quest fail, they will be forgiven. And when lost, they'll find another way. The moral imperative is the endeavor alone, whether successful or not, provided the effort is honorable and failure memorable. So if we're going to fail, we got to fail big. So just bear that in mind. And I think that's great advice for graduate students. So with that, though, moving on to unpacking my title here with consilience in disease ecology. And so consilience, this is again from E.O. Wilson, but he's quoting somebody else, is a jumping together of knowledge by the linking of facts and fact-based theory across disciplines to create a common groundwork of explanation. So it's really about how do we know truth beyond sort of, you know, the subjective. And I think that's probably topical at the moment. So in disease ecology, and we're coming from different sides, right, where there's people working in virology, bacteriology, or the ologies at, you know, small spatial scales, very detailed mechanistic work. Then there's people modeling population dynamics of various host systems and how diseases will affect those. And then there's a lot in the middle where the different people are puzzling here and puzzling there, and because there are not neat edges to put that puzzle together, you never know quite where you are with respect to each other. And when those come together, I think that's very exciting. And that's what we've been trying to do in this buffalo system. So assembling the puzzle, buffalo and foot and mouth disease virus as a model system, what I want to talk about most today is actually this intersection here on the left from the very tiny scale, virology and immune and viral interactions within the host, and then looking at whether we can infer from these tiny scales and within host scales anything about disease dynamics at the population scale, which is a reach. So, you know, maybe it'll fail spectacularly. We'll see. But in order to introduce that third arrow that I'm going to come around to in the end, I've got to tell you a little bit about arrow 1 there, so connecting the within host scale to population dynamics. And that first bit, okay, well, first study system. So the pathogen part of my study system here is foot and mouth disease virus, which is a single-stranded RNA virus. It's transmitted directly and via fomites. And so as a single-stranded RNA virus, it evolves really fast, and it's an exciting system for that reason. It's globally distributed. It has a really broad host range, cloven hoofed ungulates. It is massively contagious, which is also why it makes a good study system for studying disease transmission, because you're going to get lots of it. So the R naught has been estimated previously in cattle at 23, despite a very brief infectious period of just a day and a half or so. And so it causes these explosive outbreaks. Oh, R naught, basic reproductive number. I think after 2 years of pandemic, you're probably familiar with it. So this lets me build on this experience. It is the most trade-restricting livestock disease in the world because it's so explosive and causes massive economic losses, particularly in the developing world where FMD is endemic in a lot of places. The host part of my study system here is the African buffalo, which is the natural reservoir of FMD viruses in sub-Saharan Africa. So it's where these viruses evolved in that part of the world. There are other strains in other parts of the world that are not buffalo specific. Buffalo, it's a large ungulate. They give birth seasonally, occur in herds of 30 to 1,000 animals, and they carry a lot of diseases. It's a great model system in disease ecology because the pathogen and parasite community is so rich in buffalo. In buffalo, being the reservoir, foot and mouth disease viruses cause mild lesions, sometimes, typically none. People didn't think that they ran a fever, but they do, which we just showed in our experiments. And the transmission is really efficient in buffalo herds. So by the time they're 1 or 2 years closer to 1, I think, of age. They're all seropositive. They've all been exposed to the 3 serotypes of South African territories SAT foot and mouth disease viruses. So SAT 1, 2, and 3 occur sympatrically in these buffalo populations. So what we've done is we've collected experimental and observational data. First of all, with a cohort study of a herd of 60 to 70 buffalo, depending on births and deaths in a given year, which we captured every 2 to 3 months for 3 years. All of them, sometimes we missed a few, but mostly all of them, to monitor FMD transmission. And from that cohort study, we got some of the basic parameters: birth rates, death rates, and importantly, the rate at which maternally derived immunity in calves is lost. And then we did some experimental studies. Today, I'll just be talking about the acute infection experiment. We also did a chronic infection one, which is important for disease persistence, but today's talk is about acute transmission. So we needle infected groups of buffalo with each of the 3 serotypes, SAT1, SAT2, SAT3. We kept those in separate areas so that they wouldn't cross-transmit. Then we exposed new groups of naive buffalo to those needle-infected individuals and watched transmission occur from the needle-infected to the in-contact naive buffalo. And we used the timing when the infection arrived in our naive animals to derive a latent period, infectious period, and transmission rate for each of those viral serotypes. So those are the parameters that you need to parameterize a disease dynamic model that then describes how that translates up to transmission within a buffalo population from our little mini populations here. So it generalizes, the model just uses those parameters to generalize to larger populations. And so that work's published, and that paper is mostly about disease persistence and how that works in such a rapidly transmitting infection. But that's not my focus today. So here I'm just showing you the parameters for acute infection that we derived. And there the main take-home message is, whoa, I'm being...
Did your kids get a hold of that?
No, I just did that in the office. I'm being Zoom bombed. I don't know what that is. Does it go away when I go back? No. The scribble will remain. Interesting.
Probably the annotate.
But I didn't touch it. Okay, you'll have to overlook the squiggle. I don't know how to get rid of it. Anyway, so the take-home message from this work is that there's some pretty serious variation between these different viral serotypes that have quite different dynamics in buffalo populations, as derived from these experiments and from the observational data in that buffalo herd. So here the latent period is short in SAT1, long in SAT3, SAT2 is intermediate. Same for the infectious period. And then for the acute transmission rate that we derived. So the transmission rate and infectious period are both larger/longer in SAT1, so it transmits really rapidly and efficiently. SAT3 much slower, and SAT2 intermediate. And that adds up. You can calculate from these the basic reproductive numbers of these viruses in buffalo. And SAT1 is similar to the high estimate in cattle, so an R0 of about 24. And we did look at variation as well, but doesn't all fit into one little 10-minute talk. SAT2 is 7.8, so intermediate, but similar to SAT3 at 7.2. All of them very high R0s, right? So, you know, your SARS-CoV-2 is usually somewhere between 2 and 3, and maybe Omicron is a little higher, but these are very high. But very variable among viral strains. Previously, these viruses were just modeled as foot-and-mouth disease virus. No strain variation. So when you look at dynamics in populations and from these parameters, what you see is pretty much what you'd expect, that SAT1 with the highest R0 has these explosive outbreaks and then peters out very quickly, whereas the others are a little bit more protracted because they transmit more slowly in a host population. This, though, is just background for the within-host viral and immune dynamics that I want to tell you about. And that's work that's going on now that is not yet published, hopefully about to be submitted by the graduate student that's working on it. So this takes us way down to tiny scale and to what to do with data commonly collected by virologists. And so data that we collected, or my virology colleagues from the Purbright Institute in the UK collected during these experiments, looked at viremia, for example. So that is the titer of virus in buffalo serum during these experiments. The curves you want to look at are the pink ones. Those are in contact naive animals that received infection by the natural route, not by needle. The blue ones are the needle infected. We also looked at innate and adaptive immune responses. So these would be neutralizing antibodies specific against that strain of virus. We used a couple of different measures of innate immune responses. The one I'm showing here is haptoglobin. So that describes some viral biology, but what you do with it in terms of understanding how this will affect disease dynamics. Can we connect the differences in the biology of these strains to how they operate in host populations? Can we leap across scales, in other words? So we did something that immunologists would just hate, but I think is great. We modeled immune and viral dynamics. The reason they'd hate it is because it is a grotesque simplification of what really goes on in a mammalian host, right? But hopefully we can capture some of the essentials in that simplification. All models are wrong. How wrong are they? Can we capture the dynamics that result? And so what we did, don't get too carried away by the detail here, but basically what we did was make a compartmental model where we model pathogen populations P, we model innate immune responses I, and adaptive responses A. And so the pathogen grows within the host. It gets cleared in part by the innate immune response. The innate immune response acts as a spark for the adaptive immune response because the adaptive response will not just willy-nilly start making antibodies to something it hasn't seen before. But ultimately, then the adaptive response clears the virus from the buffalo. Now, note that we don't have a death rate for antibodies in here. That's because we're dealing with acute infection and that antibodies do persist for a while. Ultimately, in the long run, of course, they decline over time. So what do we get from this? We get a really nice model fit to our data here. So this shows you the data individual by individual for our in-contact buffalo, where the blue, green, and red are color-coded for the different viral strains. So that's viral dynamics in the buffalo. And then the light gray is the innate immune response, which charges up a little earlier than the adaptive, which is shown in dark gray. The adaptive immune response ultimately shuts down the viral replication. In yellow here, we've shown fever just to show that they do mount a fever. So our model captures these dynamics actually really well. So what can we do with it? So we can then go ahead, oh, the animation's a bit messed up, but what we can derive from that is we can get viral and immune dynamic parameters that describe the interactions between pathogen populations and the host response, and then maybe link that up to how well that buffalo is going to transmit virus and what its role is going to be in population dynamics of the pathogen. Okay, so that's where we're going with this. So what we can derive then are parameters for viral dynamics. So up here, I'm showing for each of the viral strains, SAT1, SAT2, and SAT3, parameters derived from the model for initial viral load, viral growth rate, and the time to maximum viral load. And so what you see there is that basically SAT1, sort of congruent with what we saw in the population level data, is massively more transmissible based on these within host parameters. So it can transmit with a much lower viral load than the others. That's the left panel there, panel A. Its viral growth rate is much higher than the other two. That's panel B. And consequently, it reaches its maximum titer really quickly compared to the others. So the time to max titer is low. We can then also look at viral fitness across these strains. So viral fitness you could within host assess as maximum viral load or as the cumulative viral load. So the area under the curve of the viral growth curve, right? So how much virus is produced by each of those hosts. That would be a measure for viral fitness in that host, right? And what's interesting there is that that doesn't follow the same pattern, right? So what we get is we get pretty comparable viral fitness between SAT1 and SAT3, despite that they have really different strategies. SAT1 is that fast growth, SAT3 is kind of laid back, but carries on for a while, right? SAT2 is the big loser in terms of viral fitness here. And what gives us a hint at why that might be is when we look at immune response parameters here from those data fits, model fits to our data. And what we see here, so it's innate activation rate in F there, adaptive activation rate and clearance rate. What we see there is that the unfortunate SAT2 activates that immune response much faster than the other two variants. And so it gets shut down much faster. So even though it has a growth rate that's not too far off of SAT1, it gets shut down right away by the host and doesn't get to make a lot of viral copies in each buffalo. Okay, so then can we make that leap across from the within host viral and immune dynamics to how that translates into populations? And this is still a little bit speculative because we really need to work with more viral strains than we did to get proper replication here, right? But each of these panels here shows you correlations between a parameter measured in the within host viral immune dynamics work, so adaptive activation rate, viral growth rate, sample mean viral growth rate, against a parameter estimated at the population scale by looking at transmission among groups of buffalo. Okay, and so what we see is that our viral growth rate variation within each individual buffalo and among strains, which is still color-coded as before, predicts really well the latent period that in part determines how fast that virus will spread through a population. The sample mean viral growth rate also predicts really well the transmission rate, which is a key parameter. And there we had to use sample means because the transmission rate was estimated at the group level, not from one buffalo to the next. We would need genotypes for the particular viral strains in each individual to pick that apart to the individual level. That's why those are sample means, and that's why the replication there with 3 is really terrible. But hopefully we'll get to work with some more viral strains and do that better. Then down here, the adaptive activation rate, which is what shut down that SAT2 so early, right? That really determined cumulative viral load and viral fitness in each individual, correlates really well with the infectious period. And when we put those all together, we can actually plot in the space of viral growth rate and adaptive activation rate, which seem to be the key parameters governing viral dynamics in each individual buffalo, how that would play out for predicting R naught. And this is very qualitative. Again, we've only got 3 viral strains here, but it's nonetheless exciting because it hints at us being able, from the behavior of a virus in individual hosts, to predict its basic reproductive number in host populations. If we can do that, that's really exciting because we can do experiments in individual hosts. We cannot do experiments across host populations, right? So if we're wanting to assess which virus is going to make it in a population, which virus can invade, these can all invade, but can they invade against other related viruses is more the question. They're all great at invading, obviously, with higher noughts. But if we can do that by assessing them in individual hosts, then we have a tractable tool for potentially predicting or at least understanding who will spread in what population. And so that's sort of on the path to the holy grail here, perhaps, or maybe once we add more viral strains, we'll fail spectacularly. This is, of course, a massive collaboration with lots of people from different places. Yeah, and that's it for today. That's Sabi. She's very non-intimidating, so you can ask questions.
So for the 2 slides back, not to go away from her, but it seems like there's a little more variation in the groups that had the green one.
You mean in here?
Yeah, like a little more spread than in some of the others. I was just curious about, so each point there's four individuals, is that right?
No, each dot is in one individual.
Oh, it's one individual?
Yeah.
Okay. So were you surprised that there was, I guess I would have, in my field, we're always interested in the individual variation in immune response to the same pathogen. So were you surprised to see like such strong separation between these 3 strains?
So we didn't really know what to expect because they haven't been dealt with in a comparative way. In fact, the SAT viruses have a lot less work done on them than the sort of Asian European varieties, A and O. So we didn't really know what to expect, and this type of question hasn't really been asked. What was immediately obvious is sort of the flip side of what you're pointing out. So you're pointing out that there's some variation in the SAT2 and also in the SAT3. What was really striking is how incredibly consistent the response to SAT1, which is really strange. What what it looks like to me that's happening there is that SAT1, when you look at population dynamics, does really extremely well in buffalo populations. When you look at sensitivity analyses, there are not many ways to improve it. It's very adapted to that buffalo host, and it's very tight in all its parameters. There's not a lot of variation, whereas SAT2 and 3, there's, I think, other things going on than just hanging out in buffalo. And so my hunch is that there's more variability there because we don't know the full story for these, whereas SAT1 just seems like the buffalo champion, and it is optimized for that host.
Yeah, okay. And it's not actually making them sick, either,
No, I mean, they'll run a transient fever.
right?
It's like a cold, sort of.
You said all these buffalo get all 3 of these before they're like 2.
Mhm.
Is there any indication of the order in which they get them in plays any part in terms of how they respond to any of the strain?
Yeah, so that's a really good question. We don't have the resolution of data to test that because calves are rarely captured when people do buffalo surveys for all kinds of reasons. I think that we should capture more calves, but we'd have to catch them pretty early to test them. And it's not totally easy because then they're still hanging out with their mothers. And then, you'd have to dart them both because, if you don't get the mom, then she'll be really angry while you're working on the calf to get some blood. So I think that would be interesting to test. The prediction, of course, would be that they get SAT1 first because it does transmit so fast. It also persists really well in each herd. It makes, what I didn't show you is that SAT1 makes carrier hosts very reliably and transmits from those without too much trouble. So we would expect annual epidemics in each herd when the calf cohort is ready for them. So when they've lost their maternal immunity. Whereas these other two either don't make a lot of carriers or transmit more sluggishly from them. So you would expect the wait period before that comes around to be a little bit longer. But we have not tested that. And then whether the order of infection and whether I've seen 1 or 2 infections before with other viruses matters. Probably not for the acute infection because there's not cross immunity between them, but for carriers, it may matter. So carriers localize virus in the tonsils. And so it may matter whether somebody's already sitting there on those dendritic cells for, the next carrier being successful. We don't know. But there's work suggesting from co-infection experiments that colleagues have done that could matter to how long those carriers persist. Yeah.
There's a question in the chat that it says, did SAT1, 2, or 3 evolve from one another, or did they all evolve independently from one ancestor of the strain?
I actually don't know. I don't know how the branching patterns look there.
I can answer that as well.
Excellent.
Sorry.
Go ahead.
SAT1 is the ancestor. It comes from an ancestral one, then branch it out from the SAT1 to SAT2, and SAT2 from SAT2 to branch to SAT3.
Okay.
From the phylogenes I saw.
Okay.
Yeah.
Well, there you go.
What do you know?
Right. Anything more? All right. Then I think it's beer o'clock.
Yeah, okay.
All right.
Thank you.
Bye, go.
You're- Unless you
Oh,
wanna take
yes.
us home.
Right. No.